Today, we're talking about CRISPR. It's a technology that lets us edit the very code of life itself. But this isn't just a story about a cool scientific breakthrough. It's about a tool so powerful it forces us to ask some of the deepest questions we have about health, about what's fair, and really about the kind of future we want to build. So here's how we're going to break this down. First, we'll get into the science of what CRISPR actually is. Then we'll see how it's being used to potentially cure real people and the shocking price tag that comes with it. From there, we'll dive into the deep ethical questions this raises, zoom out to see how CRISPR could edit entire ecosystems, and finally, talk about how we might move forward with all this responsibly.
All right, first things first. What exactly is this tool that has the entire scientific world buzzing? And what makes it such a revolution?
Okay, I know, that name is a mouthful. Clustered, regularly interspaced, short palindromic repeats. But the idea behind it is actually pretty simple. See, scientists didn't invent CRISPR from scratch. They found it in nature. It's basically an ancient immune system for bacteria. When a virus attacks a bacterium, the CRISPR system snips out a tiny piece of the virus's DNA and stores it. It's like creating a most wanted poster for that virus.
So, if that same virus ever shows up again, the bacterium uses that stored genetic memory to create something called a guide RNA. This guide then teams up with a powerful DNA-cutting protein called Cas9. The best way to think about it is this. The guide RNA is like a GPS coordinate, and Cas9 is like a pair of super precise molecular scissors. The guide leads the scissors right to the matching viral DNA, and snip, the threat is neutralized. What scientists figured out was that they could hijack this system. They could make their own custom guide RNAs to target any DNA sequence in any organism. It's literally a find and replace function for the code of life.
And this is why CRISPR was such a massive game changer. I mean, gene editing wasn't new, but the old ways were incredibly hard, expensive, and slow. You had to build a whole new complex protein for every single gene you wanted to edit. It was a nightmare. CRISPR changed all that. Because it relies on simple RNA guides that are easy and cheap to make, it made gene editing faster, cheaper, and accessible to basically any lab in the world. It democratized the technology that used to be only for the elite. Okay, so let's take this out of the lab and into the real world. Let's see how this incredible tool is becoming a source of real, tangible hope for people suffering from genetic diseases.
And there's maybe no better example than sickle cell anemia. About 100,000 people in the US, most of whom have African or Latin American roots, live with this incredibly painful blood disorder. And here's the thing that's so wild from a scientific perspective. This entire devastating disease is caused by a single letter mistake in their DNA. Just one tiny error. That makes it the absolute perfect target for a precision tool like CRISPR.
And honestly, the results from clinical trials has been nothing short of miraculous. We're talking about patients who've been in and out of hospitals with debilitating pain their entire lives, and now they're basically symptom-free. This isn't just a treatment. It's a potential cure for a disease that has been historically and systematically overlooked and underfunded, especially when you compare it to conditions that mainly affect people of European descent. It's a huge win for science for sure, but it also puts a spotlight on some long-standing inequities in medicine. But that incredible win comes with a price tag. A really, really big one. The estimated cost for this one-time cure is somewhere between $2 and $3 million per person. And that just lands us squarely at the most difficult question of all. We might have a cure, but who is it a cure for? And this is where it all gets messy, right? This is where the beautiful promise of the science slams into the complicated, unequal realities of our world. This technology doesn't just exist in a lab, it exists in our society with all its history and baggage. So one of the first big problems is the data itself. The vast majority of the genetic data that scientists use to develop these therapies, nearly 80% of it, comes from people of European ancestry. Meanwhile, the communities most affected by sickle cell are barely represented. What does that mean? It means a therapy designed with that data might not work as well, or it could even have unexpected risks for the very people it's supposed to help. It's a massive scientific blind spot. And this isn't just a what-if scenario. There's this idea in public health called fundamental cause theory. Basically, it says that big medical breakthroughs don't automatically make things more equal. In fact, without a conscious effort, they usually make things worse because it's the wealthy and well-connected who can actually access them. So unless we make a deliberate, massive effort, this multi-million-dollar miracle cure could end up making the health gap even wider than it already is. Now, we also have to talk about history, because for a lot of people, especially in the disability community, All this talk of correcting and eliminating genetic conditions doesn't sound like progress. It sounds scarily familiar. It sounds a lot like eugenics. In the 20th century, the science of genetics was used to justify horrible policies aimed at weeding out so-called defective people. And even though things have changed, that history casts a really long shadow. For many disability advocates, this feels like a high-tech version of that same way of thinking, trying to eliminate human differences instead of making room for them.
And it really comes down to two totally different ways of looking at the world. The medical model says disability is a problem inside a person, a flaw that needs to be fixed or cured. But the social model of disability flips that idea on its head. It says the problem isn't the person's body. The problem is a society that wasn't built for them. The problem isn't the person in the wheelchair, it's the building with no ramp. So the focus isn't on fixing the person, it's on fixing the barriers in the world around them. Which brings us to this huge, profound question. Is a genetic condition, like deafness or dwarfism, a disease we should try to wipe out? Or is it a part of human diversity, with its own culture and community, that we should value? The argument from disability rights advocates is that the very act of trying to eliminate these traits sends a message that the lives of people who have them right now are less worthy. This is not a science question, this is a values question.
Okay, so far we've been focused on people, but the power of CRISPR is so much bigger than that. Scientists are looking at ways to use it to edit the genetics of entire species, which could change our planet forever.
So, quick high school biology refresher. Normally, when an animal or plant reproduces, any specific gene has a 50-50 chance of getting passed on to its kids. It's a genetic coin flip, which is why traits tend to get diluted over time.
Well, there's a new technology called a gene drive that completely breaks that rule. It uses CRISPR to rig the game. It ensures that a modified gene gets inherited almost 100% of the time. The coin flip is gone. It becomes a guarantee. And what that means is you can make a single genetic change and watch it sweep through an entire population or even an entire species at an incredible speed.
The promise of this is just mind-blowing. We could potentially engineer mosquitoes so they can't carry malaria, saving hundreds of thousands of lives every single year. We could get rid of invasive species that are wrecking ecosystems. But the risk is just as massive. What if we accidentally wipe out a species that turns out to be a critical pollinator or a vital link in the food chain? The ripple effects could be catastrophic, and once you let a gene drive out into the wild, there's likely no going back. And then there's the truly terrifying thought that it could be weaponized. The stakes are as high as they get. So what do? We have this tool with the power to cure disease, redefine what it means to be human, and literally re-engineer the natural world. How on earth do we move forward? Well, the consensus seems to be that the answer isn't just more science, it's more conversation.
And one of the most promising ideas is a model called community-based participatory research. It's a pretty simple concept, really. Instead of scientists sitting in a lab creating solutions for a community, they work with that community as equal partners from day one. This means patients, their families, and community leaders get a real voice and real decision-making power in the entire process.
This whole approach is built on a foundation of respect. It means being transparent about the goals and the risks. It means making sure the community itself actually benefits from the research. And most importantly, it means giving the people most affected a real seat at the table when it comes to ethical decisions. This is how you build trust, especially with groups who have every reason to distrust medical research. It's how we make sure the tools we build are not just effective, but are also accepted, trusted, and available to everyone.
In the end, the challenge of CRISPR isn't really a technical one. It's a human one. The science gives us this incredible power to rewrite our own code, but it doesn't give us the wisdom to know how we should use it. That wisdom has to come from us, together, because CRISPR can change our DNA. But the real question we have to answer is this: how will we let it change our humanity?